I got some colonies, but have these colonies got my insert? Colony PCR is a way that I can quickly check if the genetic recipe that I put into the bacterial cells is actually in there. So let's take a look. Here's what it actually looked like when I was doing it this morning, and now let's talk about what's going on molecularly. In molecular cloning, you take the genetic recipe for something like a protein, and you stick it into a different piece of DNA that's easier to work with, called a vector. Often these vectors are circular pieces of DNA called plasmids that we can stick into bacteria, and the bacteria will make lots of copies of it for us. And if we want, we can get the bacteria to make protein from the instructions that we put in. But we want to make sure that the gene gets put in there correctly. One way that we try to ensure this is with antibiotic selection. So in that plasmid that has our gene, there's also an antibiotic resistance gene. And so this is in the generic part, like the backbone of your vector, not the insert part you put in. And so this antibiotic resistance gene has the instructions for making a protein that'll inactivate the antibiotic or pump it out of the cells or that sort of thing. So if you grow the bacteria in food spiked with that antibiotic, theoretically only bacteria that have taken in the plasmid will have the antibiotic resistance and will thus be able to grow. However, this isn't always the case. For example, for some antibiotics like ampicillin, the antibiotic actually gets pumped out of the cell, or not the antibiotic resistance gene gets exported from the cell, so it can be kind of shared with nearby cells. So if you grow it too long, you get these things called satellite colonies, which are these little colonies on the outside of the bigger globby colonies. And so the colonies are just the spots in the plate where a big clump of genetically identical bacteria are growing. So those colonies are growing, those little satellite colonies, but they don't have your plasmid in them. You can also have a problem where you just have that plasmid vector without the insert that you tried to put in, because that antibiotic resistance part is only in the parent vector part. So sorry if these terms are kind of confusing. I've done a post on it in the past if you need to review, but so one of the common ways that you do cloning is with restriction cloning. So you use restriction enzymes, which are like sequence specific DNA scissors, to cut the insert that you want, so that'd be like your gene or whatever you're sticking in, and then cut the vector, so the backbone part that has the antibiotic resistance gene and everything, to get the bacteria to express the gene, so make the protein or whatever from it. And so you cut them and then you stitch them together. And so since you only have the antibiotic resistance gene in the backbone part, you could end up with just that parent vector in your colonies, or you could end up with some weird misproduct thing if you have non-specific products or that sort of thing. So typically what you do is you choose individual colonies. So each individual colony represents like a single bacteria. So in transformation, you stick that DNA, the plasmid, into the bacteria. Then you plate it on these petri dishes full of bacteria food, agar plates filled with food. And then the bacteria take hold and they grow on top of themselves, like when they multiply in these globby colonies. And so you can take part of these colonies and you grow up even more to get lots and lots of bacteria. So typically in these, you use these 14 mil culture tubes and do these overnight growths in 5 mils of bacteria media, so bacteria food, liquid bacteria food. And this gives you lots of copies of the DNA that you can then purify and send for sequencing to make sure that everything went okay. But that's a lot of work and takes a while, and so sometimes you just want to get a quick idea of, did it probably work? And so colony PCR is really good for this because you don't even have to purify anything. So what you do with colony PCR is you basically take just a tiny bit of the colony and you run PCR on it. So PCR stands for polymerase chain reaction, and I did a post on it the other day, but it's basically a way to make lots of copies of a specified region of DNA. So if you make design your primers. So the primers are the places where you tell the DNA polymerase, so the DNA copier, where to start and stop. So basically you design the primers to bookend the region that you want copied. So you can do this in a variety of ways with colony PCR, but the basic idea is that if your insert is there, you'll get a specific size product. And if it's not there, you'll either not get a product or you'll get a product that's the wrong size. So in this example in the figure, you have you're copying into the insert. So if your insert is there, you're going to get a DNA amplified. The piece is going to be about the size of your insert with a little more because you start outside the insert. And if your insert isn't there, you're going to get like a really short piece or nothing at all. And so you can separate the pieces using agarose gel electrophoresis, and to see the different size bands are what you would expect. And the greatest part about this is that you don't have to go through all the purification stuff. You just seriously like stick your pipette tip into the top of the colony, mix it around a little in your PCR master mix, and then because during the PCR there's that initial like melt step where it gets really hot, and there's some like all these stuff in the PCR mixture or whatever, the cells are going to break open and there'll be enough DNA that it can sequence it without you having to go through the whole like mini prep and stuff. Yeah, so basically you take an individual colony, and so at the same time that you're doing this, you're going to start a 5 ml culture of that colony. And so make sure you cross-reference them so you know which PCR reaction belongs to which 5 ml growth. And so the purpose of doing this is that this way you have that growth going, and so if the colony PCR looks promising, then you can use that growth, and if it doesn't, then you just get rid of that growth before you like go ahead and mini prep it or anything. But this way you keep the colony PCR results and the overnight cultures like cross-referenced, and you don't have to keep going back into that plate and trying to find that same colony. So the idea with colony PCR is basically that you design your primers in such a way that they're going to amplify a piece of a specific length, and so that's not very helpful, but so there's a few different ways you can do it. You can use vector-specific primers, which are nice because, so basically these bind to sequences just in the vector, and they, so you can use them no matter what insert you put in there. But if your inserts are like similar sizes or something, it's not going to tell you as much information. But it should tell you if there's at least an insert of the proper size in there. If you have an insert-specific primer, then that's more specific that your insert is in there, but then you have to design different primers for each reaction that you're testing. The most information is if you have orientation-specific primers. So with some cloning methods, such as like when you have blunt ends, you can get the product put in like upside down or backwards. So with orientation specific primers, if the insert is in backwards, you won't get a product. So the big benefit of colony PCR is that you don't have to go through this whole mini prep step. So by mini prep, I mean there's like this whole process where you have this column method. It's pretty simplified, but it does take a while and it takes some equipment. You go through these different steps. So most of all, you have to go through that like overnight growth to get enough DNA to purify the DNA out of the cells. And so colony PCR helps you not waste your time doing this for the bad colonies. And so you just do it for the good colonies and you don't waste your time on the bad ones. So we talked about this a little bit, but with the orientation specific primers, that's where you're going to get the most information. So you have one primer that matches one strand in the vector and goes into the insert, and one primer that matches the other strand in the insert and goes into the vector. And so you'll be able to see if your insert is there and if it's in the right way. But there are also different ways to do this. And another great thing about colony PCR. So with PCR, you normally have like this master mix, which has the DNA polymerase or copier, the DNA letters, the DNTPs, and salts and magnesium and stuff to keep the DNA polymerase happy. And so typically you have this master mix with all of that stuff that's generic for all the reactions. Then for the different reactions, you have different primers. But the thing with colony PCR is as long as you're like the colonies are all, you're looking for the same thing, it's like all one clone that you're testing for, or if you're using the same vector and you have vector-specific primers, you can actually put those primers into your master mix and make like a mega master mix, so that all you have to do is put like pipette it out into the tubes and then swirl your little bit of your colony into each of the tubes. And make sure to keep track, remember? So yeah, so master mix is basically just like where you have all of the generic parts. So if you were making like cookie dough, and so you were making like peanut butter, you're making chocolate chip cookies and M&M cookies, and almond cookies, and so you make the cookie dough batter would be like your master mix, and then you would take some of the batter and you'd put in your M&Ms, and some of the batter and you put in your almonds, and some of the batter and put in your chocolate chips. And so the master mix is just a way to make all that generic part more efficiently, so you don't have to make the generic part each time for each different cookie. So it's the same sort of thing here, but our master mix has all the generic stuff for our PCR reaction. So an alternative to colony PCR is analytical restriction enzyme digest. So before we talked about how you can use restriction enzymes in the cloning step to cut your vector and your inserts so that you can combine them together. But you can also use analytical restriction enzymes. So you can also use restriction enzymes for an analytical digest. So basically, instead of making copies of the DNA, like you do in colony PCR, you cut the DNA, and so you design it so that your cut sites are like, you choose restriction enzymes that have cut sites in your insert or in the vector in such a way that you'll get different size pieces. The problem with this, the disadvantage over the colony PCR, is that here you have to do that purification and stuff. So with either of these methods, however, you're not getting the full information. You can see if like the sizes are correct and that sort of thing, but you can't tell if the sequence is actually correct. So with that, you need to send it for sequencing. And so when you send it for sequencing, it's kind of similar to the colony PCR, except that what you're doing is you're just using one primer per reaction, and then during the PCR, when you send it off, and then during the PCR, then they run it, it's like it's tracking the letters it adds while it adds it, because the letters are like fluorescently labeled, and then so when the DNA polymerase is adding the letters, then it's giving a single signal. And so when you do sequencing, here you have to make your primers so that they're actually like going into your insert, so that you can read out the insert, and you'll end up with like getting back this chromatograph with these wiggly lines that you then compare to what you would expect it to be. So that's all I wanted to go over today, and so happy cloning, everyone.